All right. Good afternoon. So now we look at the ovaries. So now I'll start by looking at the structure of the ovaries, then we'll look at some of the histological features as well. So first of all, we have two ovaries. So these are bilateral organs of the internal female reproductive system. Needless to say, they are suspended by the ovarian ligament, which is attached to the body of the uterus. Okay. So we'll start by looking at the shape. First of all, is ovoid in shape, smooth on the surface, unless you are examining the ovaries towards ovulation, then you may find some inconsistencies on the surfaces of the ovary. There might be a lump known as a stigma. We'll come to that much later on. So let's begin with the layers of the ovary. So first of all, we'll start with the surface of the ovary. So the surface of the ovary is covered by simple cuboidal epithelium. Some books may suggest simple columnar epithelium. To harmonize that discrepancy, some books simply say it is tall cuboidal epithelium. So what that means is that these cells are fat cells, which we call cuboidal cells, except that the height is much longer than the normal cuboidal epithelium. So that is simple cuboidal epithelium. That epithelium is given a name by germinal epithelium. So evidence suggests that the follicles found within the ovaries may actually originate from this layer, this superficial layer of the ovaries, then migrate into the cortex. That is yet to be proven. So that is still theory under investigation. Then immediately beneath to the germinal epithelium. By the way, the germinal epithelium is also known as the Wade's epithelium. When you're discussing the digestive system, even the lymphatic system, you came across what is known as the Wade's ring. This is a ring of lymphoid tissue surrounding the oral as well as the nasal cavities. So the same individual is attributed to this epithelium. So also known as Wade's epithelium, or we can simply call it germinal epithelium. So immediately beneath this epithelium is a layer of connective tissue, fibrous connective tissue, referred to as a tunica albuginea. Tunica albuginea, this is also present. A similar layer is present in the testis. Remember that the two organs originate from the same tissue. The testis and the ovaries, they are simply gonads. So depending on the gender of the growing fetus, they will develop into ovaries if it's female and testis if it's male. So the fibrous layer beneath the germinal epithelium is the tunica albuginea. Thereafter, we have another layer, which is a cortex. So this is quite thicker than the two layers that I've just mentioned. So as you can tell, that is a cortex. Then the central part of the ovary is now the medulla. So the medulla is where we're going to find blood vessels, that is arteries, capillaries, venules, and veins. In addition, we find lymphatics as well as some nerve plexus. So that is the medulla, the central or the core of the ovary. Right now, the reason why we're discussing the ovaries is because these are the tissues or organs of the female reproductive system that gives us the gametes, similar to the testis in males. So gametes in the female reproductive system, of course, are being found in the cortex. At the same time, they will develop within the same layer, the cortex, and then eventually exit the ovary by way of ovulation. So those are the four layers we discussed: germinal epithelium, tunica albuginea, cortex, and medulla. It's worth knowing that there are four layers. Okay. So within the cortex, we find follicles in different stages of development. So the process of formation of the female gametes, which is the ovum, begins during fetal life. So during fetal life, the process of oogenesis begins and then continues on through puberty. Okay. So let's look at the steps involved in the ovarian cycle. We discussed the uterine cycle, saying these are changes that occur within the uterus, specifically the endometrium. Now, ovarian cycle, we're looking at the changes that occur in the ovaries in relation to the development of the follicles. So at birth, we see that female are born with roughly 400,000 follicles. So follicles are different from ovum. So think of it like a nest. Then inside that nest, we have eggs. Okay, now one nest, one egg. So a follicle is a single nest plus one egg inside there. So we're saying at birth, they're about 400,000. Now, by the time the female reaches puberty, that number reduces. So we see that the follicles undergo degeneration. They die off. So by the time it's puberty, only few are available. So that is about half the number that the female is born with. So about 200 to 300, approximately around that number, is what will be left off during puberty stage. So out of those 200, then we see that 15 to 20 are stimulated every month until menopause. And then out of those 15 to 20, only one follicle will mature and undergo the process of ovulation. Let's hope the mathematics has not confused you yet. 400 follicles at birth, half of them will be present at puberty. Then we're saying every month, each ovary, okay, maybe the left ovary this month, we will recruit 15 to 20 follicles to begin the process of maturation. Out of those 20, only one will mature into what we call a tertiary or Graafian follicle, which will now undergo the process of ovulation. Then the next month, it will be another ovary to undergo that process. So you see that in a lifetime, that is from puberty up to menopause, about 500 follicles are ovulated. And only a few of those will be fertilized, which is now you and I as human beings that were fertilized and then develop as a fetus, much later on born as a human being. So let's look at those steps now involved in the process of maturing a follicle. So we're saying we are starting with a primordial follicle. So remember, these are the follicles we talked about, the 400,000. They are still primordial. They're still in their early stages of development. And we're saying only 200,000 or so are available at puberty. So a primordial follicle will develop or undergo 3 stages. From a primordial follicle, it will develop into a primary follicle. Later on, develop into a secondary follicle. Then finally, develop into a tertiary follicle. But we are saying only 1 out of 20 reaches that stage. The rest undergo degeneration. So we don't have a case where 15 or maybe even 10 follicles reach maturation. It's not possible because human beings don't, give birth to offspring like other organisms, like a pig or goat, where they can be 20, 10 or more. Just hold on. Okay, sorry for that disturbance. So I was saying only one follicle will reach the stage of Graafian or tertiary follicle. The rest will undergo degeneration. And therefore, at any given time, we expect only one ovum to be ovulated from a single follicle that has reached maturation. So let's look at that process just right now. Okay. So begin with a primordial follicle. So it's saying out of those 200,000 follicles that are available at puberty, the primordial follicles, under the influence of follicle stimulating hormone. This is a hormone produced by the anterior lobe of the pituitary gland. So there's follicle stimulating hormone that is produced there. The same hormone also is responsible for maturation of sperm cells in male individuals. It's the same hormone, but there is called interstitial cell stimulating hormone, or you can also call it follicle stimulating hormone. Because in the males, we have the interstitial cells of Leydig that are stimulated to produce testosterone, which now influences the maturation of sperm cells. So, primordial follicles, remember to say only 15 or 20 are recruited once the ovaries are now responsive to follicle stimulating hormone. And this process begins at puberty, when the ovaries are now sensitive to follicle stimulating hormone from the pituitary gland. So what we see is that once the follicles are stimulated, cells from the surrounding cortex, known as stroma cells, begin to align themselves around the stimulated follicles. Okay. So these cells form a sheath. So this sheath is going to have several layers of connective tissue cells. So connective tissue cells now begin to surround the primordial follicles that have been stimulated by follicle stimulating hormone. If you can see in this diagram, that is a primordial follicle with an oocyte inside. That is an oocyte. So as the follicle will be developing, the oocyte will also be undergoing development or maturation, if you like. So that is a oocyte, then this is a follicle. Now, each primordial follicle is surrounded by a single layer of flattened cells, what you can refer to as simple squamous epithelium. So these are flattened cells. We can call them as follicular cells. Then the connective tissue cells that are now forming various layers around the follicle, we are calling them as stroma cells. These are coming from the cortex, the stroma cells. Okay. So at this stage, we are going to have two different types of primordial follicles. Okay. We have a primordial follicle surrounded by follicular cells, which are flattened. As the follicle responds to follicle stimulating hormone, these flattened cells change their morphology and become cuboidal cells, or what you can call fat cells. These cells are fattened, meaning the height as well as the breadth is equal. So all that morphological change is influenced by follicle stimulating hormone. So if you can compare this follicle to this one, you see that there's a difference. Here, the follicular cells are flattened, but there the follicular cells are cuboidal in shape. Then the number of layers of the stroma cells around the follicle also is increasing. Excuse me. So those are the two types of follicle we have. The initial follicle, which was surrounded by simple squamous epithelium, and the developing primordial follicle, which is now surrounded by simple cuboidal epithelium. Okay, so all that is as a result of influence of follicle stimulating hormone. Okay. So let's look at the primary follicle now. So the primary follicle at this stage has simple cuboidal epithelium, different from the primordial follicle, which had simple squamous epithelium. Remember that all epithelium rests on a basement membrane. So the basal pore of these cells is resting or sitting on a basement membrane. Okay. So that will be a very important feature as the follicle develops, because that marks the point of demarcation between the follicular cells and the stroma cells. Okay. So at this stage now, we're calling it as a primary follicle. And what is inside there now is a primary oocyte. Inside the nest of a primordial follicle is an oocyte that has developed from what we call oogonia. So oogonia, those are underdeveloped oocyte. Now, in the primary follicle, we have a primary oocyte. So you can see the difference in size. The size of the oocyte, the primary oocyte, as compared to the size of the oocyte in the primordial follicle. Okay, just hold on a bit. Where are these guys? You brought back, huh? Okay, that's good. All right. Okay. Yeah. All right. So we're looking at the primary follicle now. So primary follicle, the follicular cells are now simple cuboidal epithelium, of course, surrounded by stroma cells, which are coming from the cortex. Now, the primary follicle, apart from having cuboidal epithelium, these cells undergo mitosis. So these cells increase in number as the ovaries continue being sensitive to follicle-stimulating hormone. So the number of follicular cells begin to increase, as you can tell, between a unilaminar primary follicle and a multilaminar primary follicle. So still the cells are cuboidal, but the major difference is that now the number of layers is more than one as the follicle continues to develop under the influence of follicle-stimulating hormone. I'm busy hapa nuasa. Okay, so that is a primary follicle. So primary follicle, like I was saying, there are two types now, a unilaminar one and a multilaminar one. But still, if you look at the shape of the cells, these are cuboidal cells. But the multilaminar one has more than one layer of cuboidal cell, and then the unilaminar one has a single layer of cuboidal cells. Okay. So at this stage now, these cuboidal cells are no longer being referred to as follicular cells. You can come in. They are now referred to as the granulosa cells. Okay, so granulosa cells, those are follicular cells that have just increased in number. And then the cells of the stroma, these cells the connective tissue cells that were coming from the cortex, are now referred to as a thicker folliculi. So this is a thicker folliculi, if you like. So that is a thicker folliculi. Then this is a granulosa cells, which are simply cuboidal cells. Now, of importance is a layer of glycoprotein that is developed between the oocyte itself and the granulosa cells. So you can see that these are cuboidal cells with multiple layers, and this is a basement membrane. That is a basement membrane. Then there's now a layer of glycoprotein that accumulates between the oocyte that is developing inside this follicle and the granulosa cell. So we call that as a zona pellucida. You see that darkened line? That is a layer of glycoprotein known as zona pellucida. Okay. So that acts as a point of demarcation between the developing oocyte and the follicular cells, which are now being referred to as granulosa cells. This structure, much later on, we will see that it is important in the process of fertilization. Okay. For now, what I want you to know is that there's a layer of glycoprotein separating the granulosa cell from the oocyte, known as the zona pellucida. So that is a primary follicle, differentiated from the primordial follicle. Okay. I'm in class, you want to it's a simbe. Anyway. I wanted you to see these guys, but all right, it's fine. The same one. All right, now let's look at the secondary follicle. So saying the primary follicle, inside the nest, the egg there is referred to as a primary oocyte. That is the primary oocyte. You can see that now the cell becomes more distinct. Even the nucleolus becomes so prominent. Okay. Let's go on to look at the secondary follicle. So the most important feature of a secondary follicle, and this is what would differentiate it from the primary follicle, is accumulation or presence of fluids within the granulosa cells. So these are cuboidal cells. Okay, now these cuboidal cells produce some serous fluid, and this fluid begin to accumulate among the cells and create spaces. Okay, so you can see the antral or cavity. These are cavities referred to as the antrum. So at this stage, these cavities have not yet fused to form a single cavity. So you see multiple cavities filled with fluid being produced by the cuboidal cells of the granulosa layer. So this is a feature that is now distinguishing a secondary follicle from a primary follicle. The rest of the features are the same. We see that the thicker cells are still present, and they formed a number of layers around the developing follicle. And these cells further distinguish themselves from each other. You can see that the cells in the inner layer of the thicker are cuboidal in shape, whereas the cells, these are connective tissue cells, by the way, the connective tissue cells in the outer layer are flattened. As a result, the thicker follicule can now be divided into two parts, the inner layer and the outer layer. The inner layer is what we're going to refer to as the thicker interna. You can see that with cuboidal cells. Then the outer layer is what we call the thicker externa with flattened cells. Okay. At this stage, the oocyte continues to develop and increase in size. So we can now refer to it as a secondary oocyte. So that is a secondary oocyte inside the nest of a growing follicle. Okay. So these cells, the granulosa cells, continue to produce the serous fluid, and this fluid continues to accumulate within the layers of the granulosa cells. Later on, these small cavities will fuse and form a single giant cavity known as the antrum. So before formation of the antrum, we see that the thicker interna and the thicker externa receive blood vessels from the medulla. Remember that the medulla is the core or the central part of the ovary. Okay. And that's where we find blood vessels, lymphs, and nerve plexus. So blood vessels from the medulla enter the thicker cells, both the thicker externa and the thicker interna. Okay. Once vascularization begins, the cells of the thicker interna, these cuboidal cells, begin to produce a hormone known as estrogen. So estrogen begins to prepare the uterus in an event that fertilization occurs. Okay, we discussed the stages of uterine cycle. So I would refer you back to that discussion that we had. Those stages of development of the endometrium: menstrual phase, proliferative phase, and secretory phase. So proliferative phase is being influenced by estrogen being produced by the thicker interna cells. So at this stage, the follicle is now considered as an endocrine gland and is now producing a hormone known as estrogen. This hormone diffuses in the blood vessels that have penetrated the thicker externa as well as the thicker interna, and it's those blood vessels that collect the estrogen and transport them to the uterus for preparation in an event that fertilization occurs. Okay. Away from the secondary follicle, at this stage, the follicle is still being sensitive to follicle-stimulating hormone. So I mentioned of cavities within the granulosa cells. Now those cavities merge and form a single giant cavity known as the antrum. So as you can tell from this diagram, only one cavity is present in the follicle now. At this stage, we call it a mature or simply the Graafian follicle or preovulatory follicle. So this fluid will continue to accumulate as long as these cells are alive, the granulosa cells. They will continue producing this serous fluid. Another important feature in the Graafian or mature follicle is some of the granulosa cells detach from the rest of the granulosa cell, of course, being separated by the antrum, and surround the oocyte. So we call those cells as corona radiata cells. Then the point where these cells merge with the granulosa cell, we call it the cumarus oopharus. That is the cumarus oopharus. And then around it, we have the corona radiata. Same cells. It was still the same cuboidal cells that were dividing, that began as flattened cells. So at this stage, the anterior pituitary gland is being inhibited now by the estrogen that is being produced by the thicker interna cells. So production of follicle-stimulating hormone at this stage, when the Graafian follicle matures, drops. And now luteinizing hormone production increases. So at this stage now, the mature follicle will become responsive to luteinizing hormone from the anterior pituitary gland. And stimulate these cells, the granulosa cells, to continue increasing production of the serous fluid. At the same time, it prepares the surface epithelium of the ovary for ovulation of the new and mature ovum. So what happens on the surface of the ovary? We're going to see a lump where the mature or Graafian follicle is located. Remember, all these follicles are developing in the cortex, which is a layer immediately underneath the two superficial layers of the ovary. That is the germinal epithelium as well as the tunica albuginea. So this swelling we are seeing on the surface of the ovary is known as the stigma. Okay, so that is the stigma there. And then the smooth muscle cells found in the germinal epithelium undergo some rhythmic contractions. It is those rhythmic contractions that eventually lead to rupture of the tunica albuginea together with the germinal epithelium. When that ruptures, the antrum, which is a fluid inside the follicle, forces or ejects the mature follicle together with the surrounding granulosa cells. At this stage, we are calling those cells now as corona radiata. So that is a corona radiata, the cells surrounding the mature follicle. And then that layer, the thick layer we're seeing inside there, that is a zona pellucida. We established that the zona pellucida is simply a glycoprotein layer that is being secreted by the granulosa cell together with the growing oocyte inside the follicle there. So the hormone that influences ovulation is luteinizing hormone. Prepares the ovary as well as the follicle for ovulation. So once ovulation occurs, the corona radiata plus the oocyte is picked up by the fimbriae. We discussed the fimbriae under the fallopian tubes. The distal end of the fallopian tubes have finger-like structures, and those structures hold or receive the ovulated ovum and then channel it towards the infundibulum and the ampulla of the fallopian tube, where fertilization will eventually take place. So we're saying out of those 15 to 20 follicles that were initially stimulated by follicle stimulating hormone, only one will reach this stage of maturation and eventually be stimulated by luteinizing hormone to undergo ovulation. Okay. So those are the stages under which a primordial follicle will pass through under the influence of those two hormones being produced by the anterior pituitary gland. That is luteinizing hormone and follicle stimulating hormone. Okay. What happens after ovulation? Where do these cells go to? The remaining granulosa cells and the thicker cells. Remember, the thicker has two layers, thicker externa and thicker interna. And we're saying the thicker interna is a transient endocrine organ because it is now producing estrogen and progesterone. Okay, so that is a thicker interna which is producing those hormones. So after ovulation, the remnant now, the remainder, which is the granulosa cell plus the thicker interna cells, rearrange themselves because the oocyte has gone. So this structure doesn't die or degenerate there and then. What follows next is rearrangement of the cells. So the granulosa cells plus the thicker interna cells form another transient endocrine organ known as the corpus luteum. Corpus meaning body, luteum meaning yellow. So this structure appears yellowish, and thus the name corpus luteum, which now continues to secrete estrogen and progesterone. But immediately after ovulation, progesterone production increases because now the two layers of cells, that is the granulosa cell plus the thicker cells, work together to produce estrogen, progesterone. So the amount of progesterone will be higher than the amount of estrogen. And that is in readiness for fertilization. In an event that fertilization occurs, after implantation, progesterone will maintain that pregnancy until the corpus luteum, I beg your pardon, the corpus luteum will maintain that pregnancy until the placenta can take over. So this is an endocrine organ. Similarly, the thicker antenna is an endocrine organ, but we call them transient organ because they're not permanently there. They are only seen and functional at a certain stage in the cycle of a woman. Okay. In an event that pregnancy does not occur or fertilization does not occur, this endocrine organ undergoes degeneration, where connective tissue cells begin to be deposited plus collagen, replacing these granulosa cell plus thicker cells. And you find that now production of progesterone drops. And at that stage, the color changes from being yellowish to being whitish because of the high presence of collagen fibers. And therefore, the name now changes to corpus albicans. Okay. So the corpus albicans is non-functional. It's not an endocrine gland. It's not producing any hormone at that stage. And if pregnancy did not occur, then we see now that the functional layer of the endometrium will be shed off. So those are the stages of what we call the ovarian cycle. Okay, the next process we'll look at will be oogenesis and fertilization. I think for now, we can end it here. All right.